Now it's my pleasure to announce our next presenter, Dr. Shayn Peirce-Cottler, a member of the team from the Allen Discovery Center at Stanford on systems modeling of infection. Welcome, Shayn.
Hello, everybody, and I'd like to start by thanking the Allen Frontiers Group and Kathy for their investment in our center 3.5 years ago, and it's really an honor to show you some of the things that we've been working on and talk about where we're headed as a result of this investment. So thank you very much for that. So I'm here representing the center, which is at Stanford University. I'm actually at the University of Virginia, so I'm sort of the satellite East Coast member, but everybody else is at Stanford. Our center is led by Marcus Covert, and it involves 3 other PIs, in addition to myself, Denise Monak, Casey Huang, and Jerry Morrison, who you'll get to hear about in more detail as I move through these slides. So this is a discovery center for systems modeling of infection. And the vision of our center, which is stated here, is to open an unprecedented window into infection by developing cutting-edge modeling, computation, and experimental tools to characterize both host and pathogen and integrate these into a combined systems approach. Now, Carl gave a great talk where he challenged us to think about what we are made of, and starting to think about being made of Neanderthal DNA is really interesting. I'll give you another way to think about what you're made of, which is maybe equally as startling, which is that half of the cells in your body, what you think of as yourself, are actually not you. They're bacteria. So it's a kind of a motivating reason why we want to understand how our cells interact with other cell types like bacteria, other species. So when we're thinking about infection, everyone can appreciate it's a very complicated process, and it's complicated for a number of reasons. First of all, it's multiscale. That means that there are mechanisms, events that are taking place both at a spatial scale and a temporal scale that are very quick or very short, and they span all the way up to much larger orders of magnitude scale, either in space or time. So one example is that the signaling events that go on inside of bacteria, inside our cells, can happen on the order of seconds. And we also know that the cell behaviors that those molecular signaling events can elicit can happen on the order of minutes or hours or even days. And then ultimately, the colonization of our gut with bacteria is something that happens over a lifetime. So that time scale is very broad. Similarly, the spatial scale, so events can happen in the order of nanometers, microns, centimeters, all the way up to the level of meters if we're thinking about the length of our GI tract. And so coming up with tools to integrate across these scales is really a challenge, not just from the standpoint of making measurements, but also kind of bringing those measurements together to understand how mechanisms move across these scales. So the other aspect that makes studying infection hard is the fact that these processes are very dynamic. So here's an awesome movie that was provided by Denise, one of the PIs of our center. And these are immune cells in green that are being attacked by Salmonella, which is the little red dots you'll see. And what's really interesting is you can see it's starting to happen now. Some of the Salmonella has actually entered into the immune cells and then proliferated substantially to the point where they completely taken over and killed those cells. And that's kind of the red eruption that is happening there at the center. What's also interesting about this movie, which represents the third reason why infection is difficult to study, is because there's heterogeneity, which was mentioned previously in the last talk. So there's some cells in the same field of view of this microscope that are not being infected, right? And so there's definitely some differences at the cell-to-cell level that's affecting the susceptibility of infection. And so trying to understand the multiscale nature, the dynamic nature, and heterogeneity is one that's quite challenging and critically important if we're going to come up with new ways to combat antibiotic resistance, which is really what the bugs are doing all the time to become, they're evolving to become resistant to the drugs that we have. So there's a number of different antibiotic targets that we're able to use molecular chemistry to try to focus on developing new drugs to target, such as manipulating the cell walls of these bacteria, affecting the DNA synthesis, dealing with metabolism, trying to mess around with protein synthesis of these bugs. But at the same time, these bugs are working against us, so they're evolving mechanisms to keep our drugs from working. So they have efflux pumps for antibiotics, for example. That's something I'm going to talk about at the end end of my talk. So as we add antibiotics to a culture of bugs, there's a subset of bacteria in that system that are very rapidly upregulating genes that cause efflux pumps to be expressed, and they're pumping out the antibiotics as quickly as we're trying to deliver them. And that has really interesting effects on the survival of the colony, because the bugs aren't just helping themselves, but they can actually help and also hurt their neighboring bugs. So it's, again, thinking about this idea of heterogeneity and cell-to-cell interactions across scales that's really critical. Now, it's been widely acknowledged that sort of the golden age of antibiotic discovery is behind us. All of the naturally derived antibiotics that exist in the world today have been discovered, and with chemistry, we've been able to modify and manipulate and tack on different chemical components that will continue to be useful in clinical practice, but the argument's been made that this pace of discovery is not enough to outcompete the bugs and their ability to become resistant to our mechanisms. So it's really a significant challenge today. It's a global problem to come up with an accelerated pipeline for antibiotic discovery, and that's really the driver of our center, is to come up with new ways to integrate modeling, experiments, and computation to make this process go faster, which right now can take anywhere from 10 to 15 years at best. So again, these are the members of our team. Marcus, myself, and Casey are biomedical engineers. Casey's also a physicist, and Denise is our microbiologist and immunologist, and then Jerry is our software engineer. So it's a really integrated team. We're all working together on different aspects of the modeling, the computation, and the experimentation. And it's just been an amazingly fun and incredibly productive collaboration over the past few years. So as was mentioned before, I think we have incredible tools that the center has been generating to make measurements that are really have an unprecedented level of detail, both in terms of the resolution they can provide, but also in terms of the dynamic information. So this is just 2 cells that are in a culture system. These are white blood cells, and they've just been infected with this blue labeled bacteria that you can see here. One of the cells has actually been infected. The neighboring cell is still protected, but what we're able to do is image different signaling molecules. So NF-kappa B is a transcription factor that's very important for upregulating different genes that are involved in cell death and proliferation. We also saw briefly JUNC, which is a kinase that's involved in MAP kinase signaling cascade, which can cause cells to undergo apoptosis. Using machine learning techniques, we can take these movies of signaling and cell behaviors, and we can extract really important information, quantitative information about where cells are, where their nuclei are. We can track them in space and time. And then we can begin to plot for infected cells and individual cells, all of this information together, to really look at the mechanisms that might cause one cell to be infected while the other cell isn't. And moreover, the types of signals that that infected cell might be delivering to its neighbor to warn the neighbor that an infection is happening. And so this work, which was spearheaded by Denise and Marcus in their close collaboration, was recently published this year in Cell Systems. So if you're interested in this, I encourage you to look up their paper, which came out in March. So this is a ton of information, and it's very nicely presented sort of as a dynamic picture with spatial detail at the level of individual cells, 2 cells in particular. But what my lab's interested in, and kind of where we fit into this collaboration, is what if we want to scale up to many cells? So what if we're talking about cells that are growing in a colony of, say, 100 million, 200 million cells living in the gut? So for that, we use a tool called agent-based modeling, and this is a type of computational modeling approach that my lab has been developing for the past 15 years. Agent-based modeling has been used in the social sciences. It's been used in ecology and epidemiology to try to understand the emergence of behaviors from individuals in a population. And so one of our first forays into modeling infection was a collaboration with Denise, where we developed an agent-based model to simulate the effects of Salmonella infection on chemotaxis of dendritic cells. And so on the left, you can see a microfluidic chamber where Denise has cultured dendritic cells, and they're infected with Salmonella, and she's tracing the migratory paths with these purple and pink lines of these white blood cells up a gradient of a chemotactic factor, which is CCL19. And so Denise gave us these images, and we then developed an agent-based model using literature-derived rules and mechanisms that have been reported to try to simulate the effect that infection of Salmonella in a dendritic cell would have on the chemotactic ability. So it's known that when bacteria infect white blood cells, they really incapacitate the ability of that white blood cell to migrate. And that's a good thing for the bacteria because it makes it less likely that they'll be hunted down and killed by the white blood cell. And so that's what this agent-based model is simulating here. But to take a step back, just to show you how this modeling approach works, I like to demonstrate it to my students using an activity that we do in class. And I tell them that they're autonomous agents, but they have to follow my rules, exhibit behaviors based on those rules. And what they'll see is, if we take a population view, is the emergence of the new patterns that are a result of their interactions. And so in the first example, I give them a very simple rule, which is that they have to identify 2 other people in the population, and they have to call one of them their shield, and their other one is their attacker, and they have to move in such a way that they keep their shield between themselves and their attacker. And when they do that, undergrads are very good at following rules at UVA. They move around, and you can see that each individual agent is making a decision following this rule. And ultimately, what you'll find is that they start to form snakes, which is an emergent pattern. So I didn't tell them to form snakes, but that's just what happened by each agent following that rule. And over here on the right, you can see a very simple simulation of this exact rule. And that's, in fact, how it plays out in our human experiment. Then I change the rules, and I tell them that instead they are supposed to move so that they're the shield to keep themselves between their 2 attackers. And when we do that, we get kind of a comical pattern that emerges, which is shown here, which may or may not be intuitive, but it's certainly emergent, and that is clustering. So all of the agents go to one location, and that's also depicted here. So this type of modeling, which is represented by this human experiment here, is exactly how we're modeling cells. We give them simple rules that are grounded in the literature based on data, and we pull them together into a quantitative framework and then allow them to play out. And from that, we can understand sort of the mechanisms, the underlying mechanisms, and how they come together to drive spatially heterogeneous, complex, and dynamic processes such as infection. So back to the example of the chemotaxis of the dendritic cells when they're infected by Salmonella. These plots are meant to demonstrate the frequency of migration in different radial directions. So when we have an uninfected cell, you can see that dendritic cells are very good at undergoing chemotaxis in a directional manner up a gradient, and that's indicated by the bars that are moving upwards. Whereas if we have infected cells, that's actually restricted, as I mentioned. The Salmonella want to prevent those dendritic cells from being able to migrate. And we've simulated this, and you can see the results on the right side of the slide here. The infected cells are able to undergo chemotaxis in a directed manner, whereas the simulated, the infected cells are not able to. And so we've been able to kind of predict or recapitulate that migration effect, chemotaxis effect as a result of infection. Now, interestingly, Salmonella, like most bugs, harbor different genes. They're called pathogenicity islands that encode for different proteins that are virulence factors. So they're sort of the ways that the bugs prevent our cells from naturally defending themselves. And so Denise has a cell line where this pathogenicity island has been genetically knocked out, and she's cultured these cells with the Salmonella infection and shown that, in fact, what you would expect is that you get a recovery of the dendritic cells to be able to migrate in response to that chemokine. And so we performed the same experiment, a simulated knockout in our agent-based model, and we were also able to recapitulate the recovery of chemotaxis as a result of the cells being infected by bacteria that lack the ability to disrupt the machinery that causes chemotaxis. So this was an exciting kind of first attempt by our group to model infection. But what we are really trying to do is extend our models to make them more sophisticated. And so a very natural coupling has happened in the center, which is to combine the whole cell modeling that Marcus Covert is very well known for doing with agent-based models. And so to introduce Marcus's models just a little bit, they are way more complex than the agent-based models that we're able to develop. He's made models of E. coli most recently, and they really do explicitly simulate many different processes inside the cell: DNA transcription, translation, protein synthesis, protein degradation, compartmentalization of proteins to different parts of the cell, changes in the cell membrane, many aspects of cell metabolism. In fact, his models, which incorporate all of the information shown here, include over 19,000 different parameters, which is an impressively sized model. There's exquisite representation of different mathematical representations of different parts of cell physiology, from growth to replication to metabolites, RNA, protein. These are integrated into a unified framework, which before our center existed required the use of a supercomputer, but thanks to Jerry Morrison, our software engineer, we can now actually run these models on our laptops, which is incredible advance. And the amount of data, the amount of predicted outcomes that these models can now give, it's amazing. It's pretty much everything you'd ever want to measure inside a cell. Changes in mass, changes in transcription rates, gene expression, amount of protein, cell division, all kinds of different things are predicted that you can actually also measure in the lab and compare head to head. So this is just a very simple sort of video to show you the output of these types of models. We can track, as I said, cell mass. We can look at the size of the chromosomes during duplication. We can look at specific genes that are being expressed in space and time, and really get an appreciation for a single cell, what's going on, given a very defined set of environments. So grown in a certain media, we can predict with a lot of detail how those cells are going to behave in terms of what's going on inside the cell. So you can appreciate then why we would want to take this type of model, which is super detailed on the inside of the cell, with data layer, parameter layer, equations, modeling layer, and then these phenomenal outputs, and actually couple them with agent-based models so that we can do this in more than one cell. And so this simple approach that we started out with and we're still pursuing is to just copy-paste. What if we just take Marcus's whole cell model and duplicate it 100 times? Well, that's possible. Pretty quickly, though, as you might imagine, the computational requirements go up, and we essentially exceed capacity after about 8 or 9 cells. And as we know, bacteria grow in cultures of millions and millions of cells, so we have to come up with inventive ways to kind of extract information from the whole cell models and feed them into an agent-based model that can then allow us to speed up computing time and also kind of make sense of the complexity in a more sophisticated and intentional way. But first, I want to show you a movie that represents kind of the copy-paste effect, and this is something that we're very proud of. You can see, if I can make this movie run, you can see these are individual E. coli shown in black. They're in an environment of glucose, which is shown in green. As they move around in this environment, they're taking up glucose, metabolizing it. They're undergoing migration. Each of these cells has all of the detail that you saw in the previous slides, which is again very powerful in terms of understanding what's going on inside the cell. But we're still at a limit. So what we started to do, which I think, again, allows us to kind of compute more quickly and maybe ask some more difficult questions, is to extract from the whole cell model certain key parameters And we do this in the form of lookup tables or libraries of simulations that our agent-based model can go to and pull from to guide very specific processes that are important, for example, in metabolism. So we have, for example, glucose uptake rates and lactose uptake rates that are defined by the whole cell model. They're put into an agent-based model, which is representing those processes. We have the transition of gene expression. So lac operon is it's critical for a cell being able to utilize lactose as an energy source. And cells can kind of switch depending on their environment. They can switch whether or not the lac operon is turned on or turned off, whether or not those genes are being expressed. So that's a dynamic process that we're able to simulate. And cells can kind of transition between 3 different states, either expressing the lac operon, able to metabolize lactose, or not, or starving. And so we've put this into a multi-cell agent-based model, and we're able to predict how these cells change over time when given an environment at high glucose or low glucose or lactose. And we can also compute how the concentrations of these carbon sources are being presented over time. And so that information then can be feeded back into the whole cell model to generate a new set of conditions that then predict uptake rates and operon transition rates. And this sort of feedback loop has been able to allow us to predict some very fundamental biology that has to do with metabolism, which is a Diauxic shift, which is well defined for E. coli. We know if we have cells growing in lactose and then we switch their energy source, we can actually see their ability to shift and utilize glucose results in a lag, which our model is able to predict. So a new direction that we're taking our models into now, in addition to looking at metabolism, is simulating these antibiotic efflux pumps, because we know that they're very important in contributing to antibiotic resistance. And so we have been inspired by a paper that came out a couple of years ago in Science, which is a very, very simplified agent-based model of cells that either have these pumps or don't have these pumps. You kind of would think that most cells would want to have pumps, but it turns out there's actually an energy cost to being able to express a pump. So if you look at a colony of bacteria at any given time, there will be cells that are expressing these pumps and cells that aren't. And so we're wanting to simulate that. Doing that with an agent-based model where we have defined, again, a set of rules which are depicted on the left-hand side. These are difficult to read, but the bottom line is that they are a set of cause and effect probabilities of sort of conditional events that will happen based on the density of cells, based on the genes that they're expressing, which are informed by the whole cell model. And what we can do is predict cell growth over time in a colony, such as you see on the right side, where we have the experiment at the initial time point, and then 6.5 hours later, after the cells have been exposed to an antibiotic for 4 of those hours. And then you see a coupled whole cell model, agent-based model prediction on the right, which is showing the population expansion of simulated bugs in these same conditions. You can see there's sort of a movie that kind of depicts what this looks like when you're looking at this coupled model. You see the bugs are growing. The red dots are cells that are not able to express efflux pumps, whereas the green dots are cells that are expressing the efflux pumps. The white squares show locations in the colony that have met their maximum carrying capacity of bugs, and then the blue is showing open space where bugs can still propagate into and grow. And so over time, you get this sort of expansion of the colony based on some of these cells being able to express these efflux pumps during a treatment of simulated antibiotics. So as you might imagine, these rules and integrating with the whole cell model require a lot of panning the literature, but there's also a lot of unknown out there. And so our group is kind of pushing forward using statistical inference and machine learning to try to help guide the formation of these rule sets. And so we've done that and I just want to show sort of an example of why it matters to have good rules. So in this case, we have 3 different agent-based model runs. In the first example on the far right, we're allowing cells to duplicate and not exclude one another based on their size. And what happens is they all essentially stack up on top of each other to an infinite amount, and that's why they're all in the center. They don't actually spread out or form sort of a biofilm. In the second simulation run, we have a very simplified criteria, which just says that each patch can only, each pixel in the simulation, this is a 2D simulation, each pixel can only hold 1 cell. And when we do that, you can kind of see there's this sort of snake-like dispersion of cells throughout the colony. And then in the third example, this is where we've used machine learning to guide the probability of proliferation based on volume exclusion. And we can see that we get a much more normal-looking colony. Everything's kind of being held together the way we would expect based on experimental observations. And it turns out that the growth rate of the population in this third case is the one that best matches the experimental measurements. So then just to wrap up, in terms of these efflux pumps, we've done simulations where we've said none of the cells can express the efflux pumps. That's shown in the second panel. We've done simulations where we are allowing 50% of our simulated bacteria to express the efflux pumps, and then a case where we have 100% of our bacteria expressing these efflux pumps over this time course of antibiotic treatment. And what was interesting to us, and sort of an emergent output we might not have expected, was that you don't actually get a huge growth benefit by being able, if all of the cells express these efflux pumps. So there's definitely sort of a nonlinear relationship between cell growth and the ability to express an efflux pump, as you can tell by comparing the last 2 simulations. So just to tie it all together, I think we're at an unprecedented point in time where we're able to make single cell measurements with exquisite spatial and temporal resolution. And now our challenge, the one that our center is really working on, is how to take those measurements and integrate them with models that can then lead us to have predictive capabilities. How can we know what's going to happen if we treat with an antibiotic or a collection of antibiotics at a certain time point for a certain population of bugs that has a unique characteristic. So the integration of whole cell modeling to get that refined detail inside the cell with agent-based models, which allow us to then duplicate that at the level of cell populations, and then of course experiments to both determine parameters needed for those models, as well as to independently validate what our models are predicting, is really, I think, the key integration that is necessary for tackling hard problems like this one, which are multiscale, dynamic, and heterogeneous, and of course play out in vivo over very long time spans and have critical determinant on world health. So with that, I would like to wrap up and thank the Allen Frontiers group one more time for all of their support. This is a picture that we took at our retreat last January at Half Moon Bay in California. It was an awesome time. We all got together, and I think the synergies that have happened, we're going to carry into the future and continue to work on these really hard problems together. So thank you.
Actually, if there are questions, please head towards the mic, just so everyone can hear them.
So that's all fascinating. So I imagine what you could do if this all works is, instead of doing trial and error in test tubes to try to find great new antibiotics, the promise would be that you could do all this in software maybe a lot faster and use ML to design antibiotics. Is that a...
Exactly, yeah, that's the vision. We've seen in our experience that computational modeling can help accelerate the discovery pipeline by allowing us to design more refined experiments, by telling us what to measure in those experiments. Maybe we would measure things that we wouldn't have known to do otherwise without the use of that model. So for sure, accelerating the pipeline. It's a lot cheaper to run these models. You could do many, many, many more conditions computationally than you can. And you also have the power of the computer, the machine learning, to help us make sense of it more than our brains can handle.
Okay, thank you so much.
Thank you.
So we're going to take a brief break. There's coffee and snacks outside, and we'll reconvene at 11 o'clock for the next set of talks. Thank you.